Hi, I'm Ella, and I am a laboratory assistant co-op at OGL. Before coming to OGL, I worked studying environmental DNA as a method for censusing pond life, and I also researched trade-offs between immunity and reproduction in termites. And both of those experiences got me really interested in the diversity of life on Earth. It is absolutely mind-blowing to think about the number of ways there are to survive as an organism on this planet, and it is so incredibly important that we are doing everything we can to preserve that diversity. That is exactly what OGL does, which is why I was so excited to come work here. We collect and preserve samples of all different types of ocean life, from microinvertebrates to fish to narwhals. I work on improving DNA preservatives that OGL uses. We want to make sure that the genetic information in the tissues in our collection is being preserved well so that scientists have access to the wealth of information that those tissues hold. I'm studying the preservative EDTA and looking for ways to prevent the DNA from leaking out of the tissue and into the liquid preservative itself. We want to be able to keep all of the DNA within the tissue so that it's available for scientists to use. I work with a range of marine taxa, but my focus has been on a couple different species of worms. I dissect the worms and store their tissue in different preservative treatments for time points ranging from 6 weeks to a year. Then I extract DNA both from the tissue and from the liquid preservative itself, and I analyze that DNA to determine how much of it has stayed in the tissue and how much has leaked into the preservative, as well as how degraded the DNA is. I am so incredibly excited to see the results of this experiment and to be playing a part in preserving the amazing biodiversity of the ocean.